Good morning, good afternoon, and good evening, everyone. And welcome to this Inside Scientific, Scientist.com, and Crown Bioscience webinar titled What's Next in Preclinical Cancer Immunotherapy Research. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be the host today. From the Crown Bioscience team, our panel consists of Dr. Gerrit Govers, Director of Immuno-Oncology, Dr. Ludovic Bouret, Vice President and Head of Research and Innovation, and Dr. Leo Price, Senior Vice President of In Vitro and Head of Crown Bioscience Netherlands. So today we're going to be discussing key topics in the field of immunotherapy, including new and emerging methods, recent publications, and future directions. So with that, I'd like to welcome Gerrit, Ludovic, and Leo to the virtual stage. Thanks so much for joining us here today. It's great to have you all with us.
Thanks, Liam.
Fantastic. Well, maybe we can kick things off with a bit of a recap question here. So why is the cancer immunity cycle relevant to the development of cancer therapeutics? So Gerrit, maybe you can lead this one.
Yeah, well, thanks, Liam. So I think indeed, if we look in the development for drugs in cancer therapeutics, by now we all know that the first line of treatment or the standard of care that we have been using for a long time is not always the solution for each indication or for each patient. And I think, yeah, it's nice that we had this whole webinar series, of course, where we introduced and looked at the cancer immunity cycle, how we can make use of this cycle to analyze different steps in this cycle that are actually subscribed are described. So we can make use of this cancer immunity cycle to better understand immunotherapies. So, for example, all the different steps that are described that are necessary for a proper anti-cancer immune response can play a role. And this cycle can basically be off, right? So you can imagine that if one of those steps is not correct anymore, that then there's no use of the immune system to basically attack the tumor cells. And by making this balance more complete again and analyzing which steps your immunotherapies can actually help, then we can make this cancer immunity cycle work and making sure to support the immune cells in killing the tumor. So during the webinars, I think we nicely described and had a look on those different steps. And we also showed which type of technologies we have available in Crown Bioscience in our in vitro capacity to actually analyze those steps. And those steps, as I said, if all of those are working in a right way, then that could lead to the right immune response that we are aiming for. So I think, during those webinars, we nicely presented those different steps, and the cycle can really help us in drug discovery for cancer immunotherapies.
Fantastic. All right, so next one here, and maybe Ludo, you can lead this one, but which new and emerging methodologies are you most excited about and why? I think this is a great question for everyone, but yeah, Ludo, maybe you can lead this one.
Yeah, thanks, Liam, for the great question. So I think there is many exciting space in the in vitro settings. So obviously, during the webinar series, we have talked about a lot of around organoids, and definitely this is a very exciting field where this has been shown that those models are very translational compared to the clinic. I would say today most of the settings is to grow those organoids in 3D matrix, but obviously the tumor microenvironment is much more complex than the single tumor cells. So there is a lot of today research and activity to set up more complex in vitro model to mimic better the tumor microenvironment. So for instance, to have vascularization and also stroma cells, which is very important, as there is a crosstalk between the stroma cells and tumor cells that have been shown to really boost the tumor growth. So actually, on the Crown Bioscience, we are working on this to make a more complex 3D models with co-culture with other cells like endothelial cells, stromal cells, and also obviously what we talk about is the immuno-oncology to incorporate immune cells in those environments. So obviously T cells, which is one of the major focus, but now there is a lot of intentions also on the myeloid cells like macrophages or also myeloid-derived cells or dendritic cells. So all those components is very critical to have a much more translational model as well. So I think all those areas are very exciting. There have been a lot of progress. Also, incorporating NK cells for cell therapy product, for instance, also is very something interesting. Also, like bispecific antibodies. This recapitulated tumor microenvironment is very critical. Also, another technology that could be applied is CRISPR for gene editing. We can actually engineer those organoids model either to understand better, I would say, the organs development, but also to mimic human disease, like to induce specific mutation, and also to understand better the drug resistance. So using CRISPR is really a great platform also to look at new target identification using CRISPR screen, for instance. And also we can use CRISPR to tag those models, so induce like luciferase or fluorescence protein. And basically we can use those models after in animal settings. So we can use organoids that will reimplant in orthotopic setting in animals models. And this is very great tools compared to, I would say, to engineering animals model that takes much more long time to generate genetic engineer mouse model. So all those, I would say, technology around gene editing also have a great application in the organoid space.
I mean, if I can add to that, I mean, I think that, Crown has aimed to make the most, to be able to provide the most translational models, and they're most well known for the animal models. And, there's this vast collection of PDX models, humanized mouse models. And that's what the most, Crown are most sort of well known for. But I think the introduction of the organoids has really given a huge boost for in vitro biology. This platform has a lot of additional characteristics which really put it above the conventional 2D models. Not only do you have a three-dimensional tissue, but these are derived from stem cells, and they've not been endlessly passaged on plastic. So it's been shown that they're really much more translational and match the original patient tumor. The other thing is that the organoids, unlike cell lines, which have been collected up from all different labs around the world, they've been grown up with all sorts of different media and using different methodologies. They're really, are the differences in responses of cell lines because of the genetics or other characteristics, or is it just because of the way that they were developed? So the organoid technology is a very standardized technology. So we have panels of organoids, about 500 of different organoids now, which have all been generated using exactly the same methodology. So then when you take a collection of organoids with different mutational backgrounds and you see different responses to therapeutics, it's much more likely that that's due to that different biology rather than the fact that someone might have used different media or something in their preparation. So the organoids, I think, provide a great step forward. And another thing I think about this platform is the availability of normal healthy tissues from the same patient, which allows you to match the tumor and the normal next to each other, gives you more of an insight into a therapeutic window. And I think that's something obviously you don't have with cancer cell lines. There's no normal tissue with them. So the organoids have been a great step forward in vitro. But I think what's really exciting now is to see how much further can we get, closer can we get to the patient. And one big step forward is really using ex vivo patient tissue. And this is something that we've been developing in Crown now for a few years and are now offering as a service, where tumor is coming directly from surgery from patients and within 24 hours going into the assay, and we're able to test therapeutics. And I think this opens up great new opportunities. There's staying much more close to the original patient tumor, both in terms of the time that it's been out of the body. They haven't had time to drift, and another very important factor is that what comes with that are the tumor microenvironment and the immune cells therein. So that offers that's really new emerging technology that's really pushing things closer and closer towards the patient.
Yeah. I couldn't agree more, Leo, with that. And what I think is really interesting is when we actually compare these two models that actually both you and Ludo just described, right? So on one hand, we have reconstituted models. So the setup is really flexible. We can make use of organoids. We can add the different players that we think are playing an important role in the tumor microenvironment, but it's in a controlled setting, right? So we can add immune cells, we can add myeloid cells, but we can also add, for example, fibroblasts or other cellular players that we think are important in actually this tumor microenvironment. So on one hand, we have these reconstituted models, and then indeed on the other hand, we have this native tumor microenvironment. So if we work with this patient material by not performing any expansion or actually thereby selection for certain cellular players in this tumor microenvironment, really maintaining the tumor microenvironment as we find it in the patient. It's really bringing those assays to a really clinically relevant assay that we can use. And what's interesting is that both of these technologies can be used in a high throughput screening platform. And I would say the high throughputness is probably even higher for these reconstituted models, right? So they're the organoids that we have, the immune cells, and we can really be flexible in the setup, but also material is in that sense a little bit less of a limitation. So we can really perform combinational treatments, screenings. So we can even do a library screen of a certain compound and really extract a lot of data out of those reconstituted models. And although this patient material that we have, we can also perform screening on that, which is really bringing much more value. And it, as mentioned, this tissue is really clinically relevant because we are really putting the patient in a disc in the lab and thereby actually performing an in vitro clinical trial. So that's really great. And both of these technologies that do have screening capacity, making use of our high content imaging technology that we have, we can also really dive into the mechanism of action, but also in the functional readouts that we see happening by using this imaging technology. So we are not looking at, for example, only one immune cell population, does it increase or does it reduce, but really looking at the complete situation as how we had it in the dish, basically, right? So either on the reconstituted or in a complete patient, we are analyzing as a whole, and thereby, using these functional readouts is really bringing much more value to hopefully in the end patients, but also to push the drug development of immunotherapies. So I think indeed having on one hand organoid technology, making use of CRISPR, this huge biobank that we can use, so materials already genetically characterized, right? So you can pick your model of interest based on certain target expression, and then on the other hand, having indeed this patient platform available and really making the translation to the clinics are really exciting new methodologies in this field of preclinical testing in the in vitro space.
Yeah, great answer. Definitely lots of technologies to keep an eye on. So a nice follow-up question to that one. How do you think these new methodologies are going to impact drug development? So Leo, maybe you can lead this one.
Yeah, I think one of the main impacts will be on improving the efficiency of selection, and I think that's going to have impact on in vitro and in vivo phases. I think with a more complex biology that we have, for example, with organoids or the patient tissues, we're going to be able to better triage compounds and make a more valid triaging selection of compounds that go into the next phase. And I think that the impact of that will be that the subsequent preclinical in vivo studies will generate a higher proportion of positive data. I think that the in vivo studies will become less and less used for selection of compounds and more for validation of the data that's already been generated in vitro, and also, of course, to give insight into mechanism of action. So I think that that kind of cost efficiency is going to play out.
And maybe also important to mention, I guess, is that in especially if we look in cancer immunotherapies, where of course there's a lot of people in the field since the development of the immune checkpoint inhibitors. A lot of people in the field are of course interested to actually see there's still limitations, let's say, in the success for patients, right, on treatment with immune checkpoint inhibitors. And actually, one of the reasons why we are still maybe lacking there is because of the right in vitro models to actually recapitulate and measure sensitivity towards immune checkpoint inhibitors. So I think there, especially the patient material that we just described, if we put this in a dish, we really observe that those patients show a similarity in their sensitivity to immune checkpoint inhibitors as we see in the clinics. So I think therefore this technology is really interesting, and really this ex vivo patient platform that we can use and actually maintaining this native tumor microenvironment seems to be playing a crucial role in still maintaining sensitivity to immune checkpoint inhibitors. And I think this was also coming out of the webinars. One of the questions asked more often by the audience, like which assays are available to check for immune checkpoint sensitivity. And I think really using patient material with native tumor microenvironment is pushing this one forward. So that's really a great development that could help for drug development in immune checkpoint inhibitors.
Yeah, maybe I can add one more comment on what Leo and Gera just mentioned. So I think indeed, I do think as well that that's going to really help to have a better selection of compounds before going in vivo studies. Both, I would say, on the efficacy, but I think also on the toxicology on those molecules. So I think that's going to have a big impact today. Most of the molecules that fail in clinic are not due to the efficacy, but more the challenge on the safety. So those preclinical approach using organoids in oncology or immuno-oncology settings, I think gonna help to address those questions and have a better refinement of the drug at early stage. That will foster, I would say, the drug development.
Yeah. I think maybe one other point that's to worthwhile mention is actually, and I think we already discussed this within our webinars, but there's also two different, and also just mentioned by Leo, but there are two different types of organoids available. So there are organoids that are directly coming from the patient derived. There are also organoids that we generate from our wide collection of PDX mice. And by having both organoids to use in an in vitro testing and screening for compounds, that's really on one hand, and then that could also lead to selection for indication to follow up with which in vivo models you want to follow up. So really aligning these models and first doing screening on in vitro on those PDXO-derived organoids can nicely follow up with in vivo studies. And I think recently this year, also Crown Bioscience published another paper in PLOS ONE where we actually made those comparisons and published this. And I think what's really nicely demonstrated that, for example, different type of therapies like biospecific antibodies, but also cellular therapies like CAR T cells. Those can be tested as shown in this graph, that indeed those organoid models really showed nice sensitivity upon treatment with those specific CAR T cells. So that's really, I think also will really help in other fields of immunotherapies, these new advanced methodologies that are developed.
Yeah, excellent. Great answers all. All right. So, do you think these changes are going to enable faster development of immunotherapeutics? And Ludo, what do you think?
Yeah, so as we just discussed a bit before, I think that indeed that to have, I would say, organoids or ex vivo tissue platform tissue are really gonna help to have a better understanding of mechanism of action of immunotherapies, or I would say drugs that target immunomodulation in general. So I think those platforms are going to be critical in the future to refine, as we just mentioned, drug selection before going in vivo model. So actually, on the slide that you just show on the presentation, we do see how on the left side, so the different in vitro approach that we just mentioned, organoid or ex vivo tissue platform, either for oncology or immuno-oncology. On the right side, the different mouse platform that we have today at Crown. So I think those two, in vitro and in vivo, anyway, are very complementary. They can address different questions. For instance, to focus on the different animal platform, syngenic model have been the horse work for evaluating oncology drugs, but also immuno-oncology drugs. However, this is a mouse immune system with using a mouse tumor, so the biology could be different, and there is definitely some limitation. GEM mouse model also have been largely used in the past. Some models are still very relevant and very translational compared to the clinics, like the KPC for pancreatic model. This model have been shown to really recapitulate the same feature compared to the cancer in patients. But however, it's still, I would say, the mouse biology. So Crown have built a very large collection of patient-derived xenografts. So PDX have been since here now for a long time, and have shown their translational relevance compared to cell line derived xenografts. So today we have a very large panel, more than 3,000 PDX model in different indication, and actually we have used those PDX actually to generate organoids, to generate PDX. So it's a very great platform because clients can start with the PDXO to screen their drugs, and then after move forward in some selected model to in vivo model. And the PDX can be used, I would say, in immunodeficient mice, so without immunocompartment, but they can be humanized either using PBMC or stem cells to recapitulate, I would say, partially the human immune system. So those models have still some limitation, but they can address some of the question. I think each model need to clearly understand in terms of limitation and see if they can answer what question we want to answer. But yeah, once again, I think those in vitro and vivo are very complementary and make a lot of sense to go through the organoids platform first, and then after moving to in vivo model. Also, we have done a lot of characterization of those data sets, either in vitro or in vivo, that lot of characterization that have been done, and this is mentioned actually on the right side of the slide. There is different database where a range of information are included here, like standard of care, genetic information with some sequencing data, looking at gene expression, mutation, but also proteomics data. So there is a huge number of information are available for all clients that can help on the selection of the model.
Yeah, great points. All right. Well, I think, great question here about AI. I think we'd be amiss if we didn't talk about that, considering its incredible rise. But how can AI be used, artificial intelligence be used, with the data being generated from these assays? So, Leo, maybe you can lead this one.
So we've already started implementing AI in our routine workflows, actually. So a large proportion of the readouts that we obtain from our in vitro cultures, particularly from the organoid cultures, are high content, and these are 3D cultures. So we have a huge amount of data, from each well of a 384 well plate. We take, maybe 100 images, and in each one of those images, there's information on the organoids that are in there, and so all of that information is extracted from the images, and we use AI tools to scan through those images and basically identify characteristics like the nuclei, the cells, the organoids by doing some pre-training. You can teach the AI tools how to do that. And so that information can be extracted, and it's accurate, and it speeds up all of our analyses. So already that's had a very positive impact. But I think there are also, there's lots more that can be done. So the data the high content data that are extracted from these images is really high dimensional. It can be hundreds and hundreds of features, and so that presents an interesting opportunity to take that complex data and put it alongside other complex omics data, be it proteomics or genomics, and start to look for interesting associations. I mean, I think in the past, if you take complex genetic data and then on the other side you have relatively simple readout, and it might be tumor shrinkage, yes or no, or an EC50, there's much less information to leverage. But with a complex data set on both sides, you can really do some very interesting analyses and extract more information and make more interesting associations. So I think we're just getting started in Crown with our use of AI to analyze our data, make most use of the data as possible, and then also to link that up to other publicly available data, and then also our own internal databases of sequencing data, for example.
Yeah, and also to add to this point, Leo mentioned, and we have a huge characterization, so all those information can be indeed used to either look at, for instance, biomarker discovery, and we have also other supporting platform that can be used where we have proteomics, so we can also do some deep proteomics and phosphoproteomics analysis combined with genomics, so all multiomics approach combined with the phenotyping, characterization, and also linking with in vivo data. So all those data set can be indeed used to have more deep dive in terms of stratification of response for some drugs, or also, as I mentioned, on biomarker discovery.
Brilliant. Well, I think to wrap things up, we'll ask you three, but we'll also ask the audience. What do you believe are the greatest developments for preclinical testing of IO therapeutics? And Kara, maybe you can lead this one.
Yeah, I think there are a lot, but I think one of these that we have been discussing multiple times now during the webinars and also here today is the organoid technology. and not only the organoids directly from the patient, but also, as mentioned, the organoids that are derived from the PDX mice collection that we have, and using those organoids in try to reconstitute basically the tumor microenvironment. So really performing co-cultures together with immune cells from different subsets, including potentially also other cells like stromal cells, and yeah, really try to mimic basically the tumor microenvironment of the patients in vitro, and also because as just mentioned, that all the data that is already available from these organoid models can also really help to, on one hand, first identify which models are the best to use for your specific um, uh, potential drug development that you are trying to achieve. But on the other hand, really making use of this tumor microenvironment and to complete it. So I think organoid models are really really exciting development in the field.
I think that my vote would have to go to the ex vivo patient tissue testing. I think, I think it's a very exciting development. We could consider it the sort of the next generation of in vitro testing. And I think, that for a couple of reasons. I mean, one really important one is that these tissues in the assays, they're sensitive to immune checkpoint inhibitors, can actually induce enhanced killing with these drugs. And that's something that's not been recapitulated, as far as I'm aware, in any other in vitro assay. So I think that that's a really important step forward, that in this, the fastest growing class of therapeutics, that we do have some in vitro assays that can be used to to test those. And so I think that, in my view, I think that that's a great step forward. And I think, where we're going to go with this patient tissue is also exciting. I think it can be applied to clinical trials. It can support clinical trials. And, once we've, for example, tested a new potential therapeutic in this assay, and we can show that this therapeutic's able to discriminate responders and non-responders, that this can actually be used to help in the selection of patients for clinical trials. These clinical trials are brutally expensive, and so in the early stage, if you just want to prove that this compound can work, if you can use this assay to actually select the patients and enrich that population, then you can get an insight whether the therapeutic is actually going to eventually can work in a population with a much smaller trial. So I think that's going to have some big impact on reducing clinical trial costs, I would think.
So in my view, what is really interesting is what organoids or ex vivo tissue platform brings in terms of getting an in vitro translational platform, which can really help to fast track and foster the drug development, and particularly it's almost impossible to test a multiple hundred of different combination which in vivo. And actually this is also what is happening in clinical trial, where there is a lot of different combinations that are tested, but I think that could be then better defined and refined at early stage, particularly in vitro. Let's say selecting the few combination that show the most promising results, and then after move to in vivo and select those ones to go in the clinic. So I do think really that's gonna help in terms of reducing the attrition seen in clinic in terms of efficacy, but also as mentioned before, also in terms of safety, where all those questions can be addressed early on.
Excellent. Well, yeah, Ludo, Kara, and Leo, thanks so much for your insights today. It's been really a pleasure having you with us.
Thanks, Liam. Thanks, everyone.
Yeah, definitely. Big thanks to everyone for joining us today. And so, yeah, in closing, we hope you enjoyed this Crown Bioscience Inside Scientific and Scientist.com webinar. And we look forward to having you with us again soon. Have a great day, everyone.